I wanted to bring this slide to you. This is just a glimpse of viral selection that many of you may think that why this virus was so successful. Then we collected a lot of viral genome from various states as well as other countries. And then we looked at just the simple Tajima-SD test to them. And then we found that all of the regions, they are strongly positively selected. This is why a very strong positive selection was working with the viral genome. And this is why the virus was quite successful in various parts of the globe. Then, while working with this, we took three major things. One was host susceptibility, which I'm going to explain here. We also looked towards the various virus variants. And then we have also done surveys and surveillance with the population. But that we are not going to discuss here, mostly host susceptibility as well as the various virus variants which we studied here. So in COVID, usually what we see that the people who have comorbidity and people who have very high age, they are mostly most susceptible to these diseases. But in some cases, we see that even younger people, unfortunately, died because of that. And some of the people, nothing happened to them with all of the age groups. So this was our major question that we started to look. And if we look to the various waves, there were three major waves in India. So the first wave was smaller, and the region was because there was strict early lockdown. The low virus infectivity was there, and public was very serious during the first wave. But during the second wave, there was no lockdown at all, and it was not needed at all. Even in the first wave, the lockdown was not needed. The virus infectivity was very high because it was mostly with the Delta and Alpha variant, as well as some cases were also related with the Beta variant. And in third wave, there was no lockdown as well. Then the virus infectivity was higher, and the most important thing was during that time, India has already acquired the hybrid immunity, the immunity which was coming from the infection as well as from the vaccine. And you can similarly see the number of deaths, which was highest for the second wave. But in third wave, the infection was nearly closer to the second wave, but the number of deaths was lower, and the region was hybrid immunity. And you would be amazed to know that we have done a survey after the second wave, and you could see that two major groups of people, they either relied on fake news or did not take it seriously. But this is not just one case. This is a very common trend what we observe in any of the situation in India. That people, they are confused and they are relying on fake news, and also many of the things they usually did not take it seriously. So our study in CCMB that took nearly 8 to 10 years, where a lot of sample was analyzed, and this was again thanks to Dr. Lalji Singh. His way of mentorship and his way of collecting sample was really incredible, that he contacted to all the smaller colleges and universities. They were bringing two of the students from various regions of India, and these two students usually bring the local population sample, and this is how CCMB so far has collected more than 40,000 samples from all over India. So we during that time we analyzed nearly 20,000 samples, and those sample has helped us to resolve many problems. One of the case was like Ajnala massacre, that is near to the Amritsar. Second case was related with the Queen of Queen Ketevan of Georgia. So those cases we were able to solve only because of the a huge database, and that was because of Dr. Lalji Singh. So based on that, we constructed the ancestry of South Asia, and there were three different kind of ancestry. One was the indigenous Indian ancestry, this green color. One was associated with the West Eurasian population, and third was associated with the East Eurasian population. So based on these three ancestry components, we inferred the ancestry of the population of South Asia. So when the COVID started to spread, we took this question that whether it was already known that the entry receptor point for coronavirus for the human was ACE2 gene, and that was existing at the chromosome X. So what we have done, we wanted to know whether the structure of this gene based on different mutation, by studying different mutation, whether it may affect the any disease predisposition. So some of the studies has been already performed by other groups. We took the Indian sample as well as the neighbor samples. So then it was the genetic structure of this particular haplotype was not studied among the South Asian population. So what we have done, we have used the whole genome data that was coming from Estonian Biocenter, nearly 400 samples. So we took this next generation sequencing data, and we extracted the 1000 as well as we took this 393 sample from Estonian Biocenter genome data as well as 1000 genome data, and then we extracted the AC2 genes from both of the data set. And then by using the phylogenetic tree construction, we constructed these trees. So usually what we see in genomic ancestry studies, that usually the South Asian population, usually they fall in between European as well as East Asian population. So they are more closer to European and distantly related with the East Asian population. So we wanted to see whether this particular gene, how this gene is behaving for the South Asian population. So one of our student, Dr. Ansika Srivastav, now she's in US. So she started to do the phylogenomics related with the AC2 gene. So here, surprisingly, what we have observed that the South Asian population was closely associated with the Southeast Asian and East Asian as well as Siberian population. So irrespective of the relation that we observe here with whole genome analysis, that South Asian was more closer to European. Instead of that, South Asian were more closer to the East Asian population. So as well as when we have looked towards the diversity, we see that there is a moderate diversity of this gene for the South Asian population, but the Southeast Asian population was showing nearly a fixation of this gene. So probably there was some positive selection, which later one of our students, Rudra Kumar Pandey, has discovered that this is because of the strong positive selection of this gene into the Southeast Asian population. So then we wanted to see that how many usually the major haplotype exist in this global population for this particular gene. So based on that, we constructed a phylogenetic tree, and here we found three major ancestral haplotypes. And here one can see that the European population, they are mainly falling here in the haplotype 1, whereas haplotype 2 and haplotype 3 are mainly associated with the Southeast Asian as well as Siberian population. And we could see that majority of the Indian samples, they are falling here. So here in absolute number, absolute percentage we calculated, and we found that nearly 60% of the South Asian individuals, they were falling in this haplotype 2 and haplotype 3. And we found this mutation RS22 or 85866. This mutation was probably reducing the susceptibility in the population. So based on that, we looked the real number of cases as well as case fatality rate among the Indian population, and we found an inverse relationship of this haplotype against this case fatality rate. So wherever the case fatality rate was high, the frequency of this haplotype was lower. So, for example, like in Northeast India, the frequency of this haplotype was high, but in Northeast India, overall, even at today's date, the case fatality rate was lowest among all other Indian states. So, based on that, we also studied two other genes, TMPRSS2 furin. Rudra Kumar Pandey or another PhD student has studied these two more genes. And there we found a very different type of correlation, where we see that the South Asian population were either falling with West Asian population or falling with the European population, as we expect in natural condition. So based on that, we modeled South Asian population susceptibility in between East Asian ancestry and European ancestry. So because of these three genes, we finally see that the susceptibility of South Asian population was closer to East Asian population as well as the European population, but it was in between. So this is why also in if we start to calculate the case fatality rate, then we see that this is lowest in East Asian populations. Then it was second lowest for the South Asian population, and it was highest with the European related ancestry populations. Then there was another study that was done by the Svante Pabo and his colleague Hugo Jaber. They have compared them the Neanderthal population with the 1,000 genome samples, the NGS data, and there they found that there was a 50 kb of genomic segment. This 50 kb of genomic segment was showing a very high level of association with the susceptibility into the global population. So this third chromosome 50 kb gene, they suggested that the human population has inherited this particular gene from the Neanderthals, and this is making people more sick. And overall, if you look to the geographical distribution of this Neanderthal core haplotype into the global population, one could see that it is not at all present among the African as expected. Also in Southeast Asian and other East Asian population, it was nearly absent. But in India, as well as in Bangladeshi population, it was the highest. So according to their study, because this population was having the highest level of susceptibility, and this highest level of susceptibility is because of this 50 kb region that we introgressed from the Neanderthals. But in reality, the real picture was quite different, because Europeans were more susceptible and Indians were less susceptible. So based on that, our student Prajwal Pratap Singh has started to look whether this gene or whether this particular 50 kb segment is making any closer correlation with the number of case fatality rate as well as number of infections over the India. Then we found that the ACE2 gene was quite highly associated with the different timelines of Indian infection. But the 50 kb segment, what they have suggested, was not associated with any kind of case fatality rate or any kind of population dynamics over the time. So our comparison has clearly showed that this allele is not at all affecting the Indian population, as well as the allele what we have, the haplotype what we have discovered was clearly associating with this with the Indian population structure. So here we compared both of them, and then we showed that this 50 kb segment was not associated with the South Asian population, at least. Then there were multiple studies which have been done on the Brazil as well as other South American population. So based on that, they showed that the isolated population, people who are isolated, so Amazonian, the tribals population, they are mostly isolated, but whenever COVID reached there, there was a very high level of death which has happened there. So based on that, we also wanted to see how the isolated population of South Asia we have in such cases. So as we recall that the major part of the language groups which South Asians speak is associated with the Indo-European language family. The Dravidian, they are restricted towards the southern part, as well as some of the pockets they are present at the central India. Then there are multiple pockets of the Austroasiatic family. Then at Himalayan fringes, there are Tibeto-Burman population. But also there are the Andamanese population who have a very distinct language. They are genetically as well as linguistically isolated. Then there is another population in Central Asia called Nihali that is linguistically isolated. Then another population in Nepal called Kusunda. They are also linguistically isolated. So we studied this population based on the runs of homozygosity. We wanted to see that how much homozygous a particular population is if we compare the whole Indian population history. So there we see that there are multiple even the Dravidian speaking population, there are Nicobaris, there are the Parsis, there are Jews, even the Himalayan Chengpa population. But the Onges of Andaman and Nicobar Island, it is showing the highest level of runs of homozygosity among the Indian population. So if segments are homozygous, means the variation in the population would be quite low, and if variation would not exist, then everyone may have the similar kind of alleles or similar kind of haplotypes. So if these kind of observation would be present in a population, then whether that population would be in high level of susceptibility or not. So based on that, we compared the Jaravas and Onge with the other linguistic groups of the India. And we found that the Jaravas and Onges, they are showing a very high level of susceptibility against the COVID-19. So we published this result and then informed the government of India that these if COVID reaches there, then it would be very difficult because there are at the moment 400 Jarava people and 126 Onge people. So if even someone would catch the COVID, then it would be very difficult to save this population. Then during COVID time, they even stopped the medical people to go and meet the Jarava and Onge people. They kept them completely isolated. And this paper was one of the best paper of Reader's Choice in Genes and Immunity Journal. So overall, what we see that the haplotype analysis for the entry gene, that is a C2 gene, that has shown that there is a very excessive sharing of the haplotype of South Asian descent is associated with the East Eurasian population rather than West Eurasian population that we frequently see. And also the possibility that this SNP is being associated with a protective role against COVID-19. And this protective haplotype was lowest among the Maharashtra state and highest among the northeastern states of India. And the Neanderthal haplotype that was suggested by Svante Pabo's work, it did not show any severity association among the South Asian population. And the Andaman Islanders, they are at the highest risk of the COVID-19, and this was the region to keep them isolated during that time. So our second work was mostly focused on the virus variants. And in one of our study, we looked to the alpha variant distribution among the various parts of the states of India. So we found that a region of India was showing a very strong surge during the entry of this alpha variant, and this distribution was showing a very beautiful pattern, and that was showing a very rapid expansion. So this founder effect was because the people were meeting frequently, and this is why the number of infection has increased exponentially in this region. And then with the collaborators in Bangladesh, we compared the CRP as well as the D-dimer levels, and there we found a perfect correlation with the high level of both of these values. And lastly, we have also done the wastewater surveillance. This was actually the best way to find out the outbreak. So whenever there is an outbreak, usually we see that the early detection of any outbreak can be easily done with the wastewater surveillance. Just bringing the samples, then doing the RT-PCR or sequencing, then can be easily could be done with such kind of practices. So this was some of our publications, and because this needs a huge amount of people to work together, and we were fortunate that a lot of people they associated with such work, and finally 75 people from India, they worked together to bring such kind of work that was showing quite a good input related with the contribution of India in COVID research. So this was it. If there will be any question, I'll be happy to answer.
Any questions? Anybody wants to ask a question? Okay, I think I will start. Yanashwar, I have a question actually on. So we have seen some disproportionate COVID incidences in various states. Like, if I remember the data correctly, the Kerala was actually, if you look at the population versus number of incidences that has Kerala was highest. Then, and for some reason Maharashtra was highest. So, do you think is it because of the genetic diversity or related to genetics, or is it more mobility in some of the states like Maharashtra and Kerala that was instrumental in?
Exactly, it could be both. So the factors, what we see that the genetics was one of the player as well, but mobility was even a bigger player that was making the more people sick or more people infected because of the frequent meeting.
And I will add one more question to that. So you looked at the very targeted, like two SNPs, which were shown to be susceptible for COVID, and then also ACE2 gene, and then there's a furin gene that you're looking at. So have you ever looked at the whole genome data itself and see like if there is any factor that any other candidate gene that may be which is not reported and is involved in susceptibility?
Recently, Rudra has completed a genome-wide study where we have used nearly 250 samples. And there we found nearly 15 to 20 genes which are playing with quite a stronger effect size and showing their association with the susceptibility.
Any other question? Anybody else? Any?
So good evening. I just have one question. How the variation in H2 sequence led to the variation in susceptibility in different populations?
Could you please repeat one more time? I did not hear it clearly.
So how the variations in H2 sequence resulted in the variation in susceptibility to SARS-CoV-2?
How the?
Ganeshwar, it seems like what he's asking, like based on sequence, do you know what is the mechanism like?
What's the mechanism? Yeah.
Any indication of mechanism?
Mechanism we did not study, but I think that because the the receptor point was in cases when there are such mutations which were blocking the entry and the association with the viruses was possibly one of the region that if the association, if they would not associate with it, if they would not bind with that, then there is no particular point of making any infection.
Okay. Thank you.
ACE2 is a receptor for the virus entry. So the polymorphisms in those genes may either protect you or may cause more binding of the virus. So that will determine the susceptibility. Because the ACE receptor, it is acting as a receptor for the viral entry.
So, but the people were getting
No, the severity will vary according to. So there are two different things. Like it is not the same that all who will become infected will have the same severity of disease. So the polymorphisms in the ACE gene were probably related to who will get infected and who will not. But there were other host factors which determined the severity of the disease actually. I mean, mainly response to type 1 interferon or autoantibodies to type 1 interferon, or the defects in different genes which determine type 1 interferon immunity against viruses. That was more in related to the severity of the disease.
That's not just AS1 alone. So that is why actually what. Yeah, that's what actually Ganesh was saying. So there's like another 20 genes that are currently they have found, which actually should be a cumulative action of those all 20 genes that finally going to determine what will happen. Ganeshwar, I have a question beyond this presentation. So I actually saw somewhere in the newspaper that under your leadership in BHU, you are actually collecting many DNA samples and you are creating a biorepository. So is it actually available for other researchers? Like we have few researchers in PGI who would like to have access to some of those samples, and if you could say like how they can access, is there a way they can write grants with you in collaboration or themselves? What is that mechanism there?
Yes, what was the plan? Like, even in other databases, we found out that the Ganga plain population, they are not very well represented. So, our major aim was to collect each and every group who are residing in Ganga plain. There are at least 10,000 groups who are residing in the Ganga plain. So, our aim was to collect at least 20 to 30 samples per population, and we collected nearly 50 samples per population, and at the total numbers is nearly 3 to 4,000 at the moment, our number of collection. So anyone can just write us an email and get the details of the population and get the DNA, whatever amount of DNA they want, they can get from us and then do their analysis just by acknowledging us.
And you are also open to collaborations and those kind of things, write grants
Yes,
together?
yes, definitely, definitely, yeah.
Yeah. Any other question? If not, okay. So, Ganeshwar, thank you so much. It was actually what Ganeshwar mentioned, that one of the best times that we had, scientifically especially, those were the time when we were in CCMB. Ganeshwar, his people were pretty young, I was young, and then, we were all just used to eat, breathe, drink science, and it was really, really amazing time. And as you see, that some of the associations that we built during that time as a student, that is what we are still carrying. And even after many, many years, uh, scientifically as well as personally. And this is not just about science that we are talking today, but what you are seeing is those are the relationship that actually needs to be maintained, and those are the one which needs to be cherished as a person. And in fact, actually, I'm so happy, like Ganeshwar and Deepak Rai and everybody, all of these people actually they are so successful. I mean, Ganeshwar is a celebrity. I don't know. He actually, you will see a lot of interviews coming from various newspapers and TV channels and everywhere. So even though everybody is actually such a successful stage in their career, but those relationships are still maintained, those collaborations are maintained. So I think that is one of the things that we all should think about when we are students, when we are postdocs, when we are at this age when we are enthusiastic about science. So thank you so much, Ganeshwar. And I was worried like you are going to be busy with Chhat today, but anyway, happy Chhat for you. I hope you are celebrating. So thank you again. Thank you so much.
Yeah.
Okay. Okay, thank you. So I think now we'll give you back a little bit time of your life back. So you have fun.